Our first lecture is by Dr. Ajith Kumar. He is an alumnus of All India Institute of Speech and Hearing, Mysore, and is currently working as Professor of Audiology. Professor Ajith has carried out postdoctoral research at Northwestern University. His research interests include auditory physiology and diagnostic audiology. He is serving as an editorial member and reviewer of several international reputed journals. Dr. Ajith has many international affiliations to his credit. He has more than 15 years of experience teaching in the area of audiology and has guided many PhD and master's dissertations. He has various national and international publications, funded research projects, and awards to his credit. I invite Dr. Ajith to share his presentation.
Am I audible, Sridhar?
Yes, sir. Audible, sir.
Okay. Thank you, Sridhar. Thank you, Sridhar, for generous introduction. Good afternoon, good evening, good morning to all of you, wherever you are in your time zone. Hope all of you are doing safe, keeping safe, taking care of your health and your dear and dear ones' health. One second. I hope all of you can see my screen. So, as all of you know, the Department of Audiology is organizing 2 half-days workshop on tinnitus and hyperacusis. You might have already seen the schedule of the program. I know that the USB or the attraction of the seminar is tomorrow. Tomorrow you have people who are pioneering in tinnitus research or who's who in tinnitus research. The person who in fact introduced the tinnitus retraining therapy himself would be talking about the tinnitus retraining therapy. The main aim of today's talks, mine and others, probably is to get everybody on a same page. As Sriraj said, we have a wide range of audience. Everybody has different levels of knowledge. Today's sessions are more or less aimed at getting everyone onto the same page so that when tomorrow the experts talk on the rehabilitation and the management of different disorders, everyone will follow what they are saying. So as a first lecture, today we will discuss about the pathophysiology of tinnitus and hyperacusis. Though the tinnitus and hyperacusis are kind of 2 different disorders, often they are combined and they are taught together, or even the research is done together, and concepts in fact are similar between the tinnitus and hyperacusis. As all of you know, the tinnitus is perception of the sound where there is no sound. The person hears the sound when no one else around him is hearing the sound, whereas the hyperacusis is person hears the sound but there is an abnormal sensitivity to the sound. So one, the person hears the sound when there is no sound. The second, there is a sound but there is an abnormal sensitivity to the sound. A lot of people consider tinnitus and hyperacusis as a similar disorder or a spectrum of disorder on a same scale. It's a continuum of disorders on a same scale. Because of this reason, a lot of concept which is true for tinnitus is true for the hyperacusis. Tinnitus is one disorder, I think, which all of us know, one disorder which probably as old as the ear and hearing existed. Because this is one condition where most of the ear disorders are associated with the tinnitus, whether it is a middle ear problem or an inner ear problem or the auditory nerve problem, or it could be any problem from inner ear to the auditory cortex is associated with tinnitus. So more than 50% of the people with hearing loss report some kind of a tinnitus. So this tinnitus has kindled equal interest in both clinicians as well as in research, because most of the clinicians when they evaluate hearing, they realize that a lot of their patients also report of tinnitus. The most common symptom, as I said, this is most common symptom for of the that is related to the ear and hearing. So, about the pathophysiology, for many years, the pathophysiology of the tinnitus is very speculative. That is, people were really not knowing how to study the tinnitus or how to figure out where is the pathology, because most often this is not a single pathology, it's a multi-form of symptoms. A lot of things can cause tinnitus, ranging from hearing loss, the ototoxicity, the noise exposure, the anxiety, anything and everything that is related to the ear or hearing, and many of it which is not even related to the ear or hearing can cause tinnitus. So because of these reasons, it was very speculative and very difficult. However, in all cases, despite such a huge heterogeneity, the final result is the same. That is, a person hears the sound when there is no sound. However, even though the reactions might be quite different from one person to another person, but the end result, that is hearing the sound in the absence of any external stimulus, is the same. So this shows that there could be some common biological basis of tinnitus, even though there is a lot of heterogeneity in terms of its origin or the causes of the tinnitus. As we all know, the tinnitus most often co-occurs with the sensory neural hearing loss. It also occurs with the conductive hearing loss, but the tinnitus resulted from the conductive hearing loss is slightly different from that of conductive hearing loss is slightly different from that of sensory neural hearing loss. In the conductive hearing loss, like for example, if the occlusion of the wax or any pathology of the ossicular chain or the tympanic membrane results in tinnitus. However, after the surgery or the medication, or if the conductive hearing loss is resolved, the tinnitus generally improves or disappears. Tinnitus generally improves or disappears. So, however, that is not the case with sensory neural hearing loss, because tinnitus generally co-occurs with the sensory neural hearing loss. The clinical evidence suggests that the peripheral hearing loss or the cochlea is the organ which may be generating the tinnitus, or which is a location of the generator site of the tinnitus, because more than 50 to 60% of the individuals with tinnitus will have some form of cochlear damage or a sensory neural hearing loss. However, it is very difficult to study these aspects in humans because the accessibility of cochlea is not all that good, and because of ethical reasons, we will not be able to do a lot of studies, a lot of invasive studies in humans. So around the 1990s, the two animal models emerged which helped us in understanding the pathophysiology and the molecular basis of the tinnitus. These two medical models were, one was the salicylate induced tinnitus, which was discovered, which was this animal model was proposed by Jastrabov himself, and probably you will learn a lot about this in tomorrow's talk also. So salicylate is a ototoxic drug which induces tinnitus in 100% of the animals or the individuals to whom it is injected. However, this is not very ecologically relevant kind of a treatment, because the salicylate is never given to the humans in such a high concentration. It's always given in very low concentration over a prolonged duration of the time. Generally in the experimental models, the salicylate is given in a very high concentration over a very short period of time. So there is a discrepancy between what really happens in the real world versus what happens in the laboratory. The next tinnitus model developed was using noise-induced hearing loss. In noise-induced hearing loss, not everyone develops tinnitus. In humans who are exposed to the industrial noise, not everyone develops hearing loss, not everyone develops tinnitus. There is a varied range of susceptibility to the noise-induced tinnitus and the hearing loss in humans. Similarly, in animals, if the animals are exposed to noise, not all animals develop tinnitus in a similar way. However, this method is more ecologically relevant because the noise exposure is one of the very common cause of tinnitus in humans. So a lot of our understanding and the pathophysiology and the molecular basis of the tinnitus arose from people who either used one of these model, a salicylate induced model of tinnitus, or noise induced model of tinnitus. Why do we need animal models? Because as we saw in the previous slide, it's very easy to control the etiology. That is, we exactly know what is the mechanism or what is the triggering point for the tinnitus. If I use a noise induced model, we know that the noise is the only variable that is causing the tinnitus. There are no other confounding variables. However, in humans, it's very difficult to control the etiology, because a person who is working in a noisy environment might report tinnitus, but the same person may be having a ear infection, or he may be listening to the music in his spare time, or he may be visiting to a busy restaurant where there is much of noise, or he may be taking treatment for some of his diseases, and one of those pharmacological agents could be ototoxic drugs. All of these confounding factors add up when we do these kind of studies in humans. However, in animals, it's easy because we can precisely control what is the etiology. Another big advantage of studying animal and interpolating the studies interpolating from animals is that the random assignment of the animals to the control group and the experimental group is possible. That is, you can selectively select animals and divide them into the control groups who don't get the treatment or who don't get exposed to the noise, or I think there are few people who are requesting to annotate the shared content. It's, I don't know what is that. I'm keep getting this request. Please don't do that. If you want slides, we can send it off late. Thank you. We were talking about random assignment of animals into the experimental and control group. This is not possible in humans because you can't select humans and divide them into the two groups and say that the one group undergoes noise exposure, other group will not undergo noise exposure. You don't have a choice. You just have to study people who are exposed to the noise. So they will come with all their predisposing factors and other confounding variables which can't be controlled. Whereas in animals, the same type of animals you can pick up, you can rear them in a very similar way and randomly assign them into the two groups, one belonging to the control group and another one belonging to the experimental group. So in animals, it is possible to use a wide range of behavioral tools starting from molecular to behavior. That is, once we induce tinnitus using any one of the two models that I said earlier, using the salicylate or using the noise, a wide range of tools can be used to assess what is the impact of tinnitus. We can use the behavioral methods to assess, and also we can use the EEGs or the ERPs or the recording of the action potential, recording of the inner ear potential, the analysis, doing the analysis of the genetic component of the proteins and the calcium structures of the inner ear. All of that is possible in the animals, whereas this is not possible in the humans. However, there are a few disadvantages, which is most often, whether it is salicylate or the noise, there is a difference between how humans are exposed and how animals are exposed. Typically, to save time, whether it is salicylate or noise, the animals are exposed to a very high doses for a very short duration. So most of the animal models explain an acute onset of the tinnitus. The tinnitus comes because the noise exposure is like, often it is traumatic and of a short duration. So which is not typical of what the humans undergo. Humans generally undergo a prolonged exposure to a loud noise, not a very high level of traumatic noise exposure, unless he is exposed to a gunshot or a cracker or something like that. Otherwise, it is most often what we see is a prolonged exposure to a loud noise or a slow dose of ototoxic drugs. So these are some differences that we need to keep while we extrapolate information from animal models to the human models. So what the models, that the tinnitus models that are developed in animals can be broadly divided into the two: the behavioral models and the neurophysiological models. The behavioral models, first, the question that people asked, the first model that was developed was whether can animal experience tinnitus or not. Because in humans, we can say that I'm hearing a sound and no one else is hearing the sound, or I'm hearing the sound when everything is quiet, or I'm hearing even when I'm sleeping, even when I'm alone in a room. But this is not possible in animals. They will not express what they are hearing, or it's very hard to make out whether they are hearing tinnitus or not. But there is no reason why animals should not be perceiving tinnitus, because animals share a lot of the auditory system which is similar to that of humans. The cochlea, the auditory nerve, the central auditory pathway, and the auditory cortex of most of mammals resembles that of humans. So because of so much of there is an anatomical similarity, there is no a priori assumption, or there is no a priori reason why animals shouldn't be perceiving the tinnitus. So if animals perceive tinnitus, then we have to develop a certain mechanism to evaluate whether animals perceive tinnitus or not, because then only it is possible to develop a model. The model can be developed only if we prove that animals are perceiving tinnitus. So in the animals, the tinnitus perception is typically assessed using conditioned responses. For example, there are two types of approaches that people have used. The first one being the conditioned responses. In the conditioned responses, what is done is an animal is trained to respond to the silence. That is, what is here the tinnitus is defined as anything but silence. The silence is not tinnitus. Anything other than silence is the tinnitus. So what is done is a low level of noise is presented in the animal cage throughout, and animal is encouraged to lick the water when the noise is present. And this silent gaps are introduced randomly in this continuous white noise background. And whenever the silent gaps are there, if animal licks the water or drinks the water, the electrical shocks are provided. So this conditions the animal in such a way that animal over a period of time learns that it has to drink water only when there is noise, and when there is silence it should not drink water. During silence, if it drinks water, it knows that it will get an aversive treatment, or it will get a punishment. So over a period of time, animal will learn to respond in this way, and it will drink water only during the noise, and it will not drink during the silent. So once the animal learns this concept through the conditioning, then the animals are divided into the two groups. One group is acts like a control, and nothing is done with the one group. In the other group, one of the two treatments is treatment, or the model that I explained earlier, is conducted. That is, the animal is given a high dose of salicylate, or animal is exposed to a high levels of noise. Now if the animal develops tinnitus, what will happen is it will be hearing the sound or the ringing sound in its ear, even though there is an absence of sound in the environment. So in presence of noise, if you introduce a silence, animal will continue to perceive its tinnitus and will not perceive the silence. So because of this, what will happen? Animal will drink water even in places where there is a silence in the continuous white noise background, because animal is hearing the tinnitus. So if this happens, then we can confirm saying that the animal is hearing the tinnitus. Once we establish that animal is hearing the tinnitus, then the other molecular studies, or other studies related to the what is the pathology in the cochlea, or what is the consequence of the tinnitus, could be studied. However, this method is time consuming, because first you need to train animals to get this kind of a behavior, or train animals to learn this behavior. Then you need to conduct this entire experiment, which would take a pretty long time. The second way of testing animals is using acoustic startle reflex. Now all of us know that acoustic startle reflex is a reflex for a very loud sound. When there is a loud sound, there is a sudden extension of upper and lower limbs, followed by cry, and there is a jerky movement of the body, which is present in both animals and in humans. Now this startle reflex originates from the brainstem, and this reflex action is used to study whether the animal is perceiving tinnitus or not. So when we give a loud background noise, there will be startle response in animals. Now, along with the noise, if we continue, if we give a gap of silence or a very low level of noise and then again give a large high intensity noise, the magnitude of the startle response decreases. Startle response in the beginning is so much, then you present a continuously low level of noise or maintain a silence for a while. After that, again you present a high intensity noise. Second time when you present a high intensity noise, the magnitude of the startle response decreases. Okay, so this is what happens normally when the animal is not having a tinnitus. So there is a difference in the startle response for the first stimulus to a stimulus which is followed by a silence or which is followed by a very low intensity noise. Now, if you induce tinnitus, if you give tinnitus inducing agents to the animals, that is either expose the animal to a loud noise or to a salicylate, what happens is the animal will not perceive the low noise or the silence because animal continues to hear the tinnitus, and because it doesn't perceive the gap or doesn't perceive the lower intensity noise, when you give a second high intensity noise, the amplitude of the startle reflex is almost same as what was earlier. So for example, in this experiment, you can see that this is experimental animal with the tinnitus. This is experimental animal without tinnitus. This is normal or sham or control animals. This is the startle response strength. So you can see that the startle response strength is not suppressed or not inhibited if the animal is hearing the tinnitus, because of the tinnitus the animal does not perceive the low intensity noise or the silent gap between the noise burst. So the startle response will remain same from the first occurrence to the second occurrence to the third occurrence. However, if the animal is not hearing the tinnitus, the strength of the startle response goes on decreasing as we go from first to second to third to fourth in this way. So if the startle response strength doesn't decrease, or if it is more than these conditions, then we can say that animal is hearing the tinnitus. So once we make sure that animal is hearing the tinnitus, then we can do a lot of other studies, like for example the effectiveness of the drugs can be studied, or the induced tinnitus is permanent or temporary can be studied. Okay, all of this is a possibility. However, as I said earlier, these procedures are time consuming, and there is a confounding variable, which is almost always any manipulation that you do to induce tinnitus is also followed by the hearing loss. So hearing loss is always a confounding factor in almost all of these studies. This is about the behavioral models. The second one is the physiological models. These physiological models depends on some kind of a physiological responses to assess the tinnitus. However, there is no direct, till date there is no direct causal link has been established between certain physiological measure and the tinnitus. When we induce tinnitus using any of those treatments that I spoke earlier, there are certain physiological change either in terms of neural activity or in terms of ERPs. All of that will happen, but all of that is necessary or will induce tinnitus is not totally clear. Okay, so with these physiological models, people have postulated certain sites for the generation of the tinnitus. These sites are not, they don't occur in isolation, because there is always a complex interaction between the peripheral and the central locations. Most often people have realized that there has to be two or more triggers to cause tinnitus. These triggers are something which are necessary to cause tinnitus. It could be incidents like hearing loss or a noise exposure or an emotional distress. Okay, any of these kind of triggers are essential to cause tinnitus, and based on this sites of generation, tinnitus has been the three types of tinnitus have been identified. A cochlear type of tinnitus, which is primarily originates from the peripheral hearing organ, which is cochlea. Peripheral dependent central tinnitus, which is originates from the cochlea, but later on it is mostly central mechanism which should maintain the tinnitus. The third one is a purely central tinnitus. So let's move on from the peripheral organ, which is cochlea. One of the portion or one of the part of organ of Corti which is indicated for the tinnitus is tectorial membrane. As all of you know, the tectorial membrane is a membrane which extends from spiral limbus and it's present over the stereocilia of the outer hair cells. The stereocilia of the outer hair cells touches the spiral tectorial membrane. When there is an excessive noise exposure, the tectorial membrane is continuously pushing, or the tectorial membrane is has been applied pressure on the stereocilia of the outer hair cells. So because of this, it might lose elasticity, or there is a continuous excitation of the outer hair cells because of the pressure that applied on the tectorial membrane. Now, the two things can happen in these circumstances. One, there is a continue because the tectorial membrane, there is a pressure applied on the tectorial membrane continuously because of the noise. It will activate the outer hair cells continuously, and outer hair cells would release the neurotransmitters continuously, and this over release of the neurotransmitters is a toxic, and that might cause tinnitus. The second one, because of the continuous pressure that fell on the stereocilia of the outer hair cells, the outer hair cells stereocilia may break up and the tectorial membrane may fall on the inner hair cells. Earlier, in the normal circumstance, the inner hair cell stereocilia were not attached to the tectorial membrane. When the outer hair cells stereocilia break up, or when the outer hair stereocilia gets damaged due to the excessive noise exposure, the tectorial membrane might fall on the stereocilia of the inner hair cells. So when the tectorial membrane falls on the stereocilia of the inner hair cells, there is a continuous excitation of the inner hair cells, and this continuous excitation would release the neurotransmitter glutamate, and that would have a cascading effect, and that's toxic for the continuous release of this is toxic to the spiral ganglion neurons, and that might damage the spiral ganglion neurons and cause the tinnitus. The second site of lesion that is indicated for the tinnitus is the outer hair cells, because outer hair cells are in almost all kinds of sensorineural hearing loss, barring a few. The outer hair cells are first affected, or outer hair cells are more affected than the inner hair cells, like for example noise-induced hearing loss, ototoxicity, the presbycusis. In most of these disorders, the outer hair cells are more damaged. When this outer hair cell stereocilia are damaged, this increases the intracellular calcium. We don't have to, I won't go to the details of that. There are a lot of ions within the endolymph surrounding the outer hair cells and the inner hair cells. One such ion which plays a critical role in the generation of the tinnitus is calcium. So whenever there is a damage in the outer hair cell stereocilia, there is a high calcium that enters into the outer hair cell. So when there is a high calcium entered into the outer hair cell, that releases the neurotransmitter glutamate, that leads to a cascading effects, and that in turn might damage the spiral ganglion neurons, as I said earlier, and result in the tinnitus. Jastreboff and their colleagues have modeled tinnitus primarily based on this fact that most of the auditory disorders, especially that of sensorineural hearing loss, causes more damage to the outer hair cells than the inner hair cells. The figure A over here shows what is normally happens during the perception of the sound. You have outer hair cells, you have inner hair cells. Now both the inner hair cells has got type 1 neurons which goes and connects to the cochlear nucleus. The outer hair cells also have a few type 2 neurons which are unmyelinated, and these are not important for the conduction of the sound to the higher center. However, these also travel and synapses with the neurons of the cochlear nucleus, and these neurons, the neurons which connects the outer hair cells, have an inhibiting effect on the neurons which is connected by the inner hair cells. So generally what happens when there is an excitation, there is also some amount of inhibition transmitted from the outer hair cells, so output of the cochlear nucleus is reduced. Now if there is a damage to the outer hair cells, what will happen? The inhibition from the outer hair cells reduce, inhibition from the input from the outer hair cells reduces. This reduces the inhibitory input from the cochlear nucleus neurons, so the output from the cochlear nucleus neuron increases. In the absence of total absence of outer hair cells, what will happen? There is only input from the inner hair cells. There is total lack of inhibition. So output of the cochlear nucleus neurons will have a very high discharge rate, which is perceived as tinnitus. This is the normal condition where you would hear perception of the sound, or the output of the cochlear nucleus is less, so you wouldn't hear any sound in silence. Over here, because of the dysfunctional outer hair cell or because of non-functioning outer hair cells, the inhibition is lost. So the cochlear nucleus neuron will increase its discharge rate because it is getting input only from the inner hair cells. Because the output is too much, now you will start hearing the tinnitus, or you will start hearing the sound which is in fact not present. However, if both are affected, inner hair cells and outer hair cells are present, both are affected, there is no output from the cochlear nucleus because there is no input from the inner hair cells. Again, you will not hear the tinnitus, and at the same time you will not able to hear even the environmental sounds or the day-to-day sounds because the output from the inner hair cell is reduced. So this theory is called discordant dysfunctional outer hair cells hypothesis. This is put forth by Prof as a main mechanism that generates tinnitus, the peripheral tinnitus, or the tinnitus that is generated by the cochlea. The next structure that could generate tinnitus is inner hair cells. Inner hair cells has inner hair cells are attached or innervated with the type 1 auditory neurons. These type 1 auditory neurons in synapses with the inner hair cells on two sides. The one is called the modular side, other one is called the pillar side. Generally, which synapses on the pillar side will have a low threshold and high spontaneous rate fibers. That's not important. What is important is the ones which synapses on the modular side, they have a receptors in the spiral ganglion cells called NMDA receptor. Now these are nothing but the receptor cells for the neurotransmitters. Glutamate is the neurotransmitter that is released by the inner hair cells. So when the inner hair cells stereocilia bend, the potassium ions and calcium ions will enter inside. When that happens, the glutamate, which is over here near the ribbons, the red ones are the ribbons. Near the ribbon, when you have a glutamate vesicles, they will release the neurotransmitter. This neurotransmitter will go and attach to a particular receptor in the spiral ganglion cells. These receptors are of two types, NMDA and AMPA receptors. On the modular side, you have something called NMDA receptors. Now, these NMDA receptors are not important for hearing because they don't work rapidly, and you know that for the hearing we need a rapid functioning because the temporal precision is of very high importance for us to understand speech or localized sound. These NMDA receptors don't have such a high temporal precision or the temporal resolution. However, when the glutamate is released, these NMDA receptors also gets activated and they will absorb the glutamate over here in the spiral ganglion neurons. But this release of the NMDA receptors, that increases ATP and the reactive oxygen species in the spiral ganglion neurons. So excess release of this glutamate, either because of the noise, the stereocilia are always open and the potassium is always entering, so the glutamate is always releasing, or because of some kind of a drug treatment, there is an excitation and the glutamate is always releasing. So whenever the glutamate releases, this NMDA receptors also will get released. So because of the overexposure, there is an excessive release of this particular receptors in the spiral ganglion cells. When there is an excessive release of these, it utilizes a lot of adenosine triphosphate or ATP. When the adenosine triphosphate, a lot of ATP is utilized, that as a byproduct, it releases a reactive oxygen species, which is toxic to the spiral ganglion neurons. So the spiral ganglion neurons will start deteriorating or start degenerating, which in turn is said to cause tinnitus. So we saw how the tectorial membrane may be a site of lesion for tinnitus, how the outer hair cells could be a site of lesion for tinnitus, and how the inner hair cells could be a site of lesion for tinnitus. However, in almost all these conditions, though these structures have some abnormality, the tinnitus is most often is caused by an abnormal firing of the action potential, abnormal firing of the action potential of the auditory nerve. Auditory nerve has two types of firing. One, stimulus related action potentials, and second, the spontaneous activity. What is spontaneous activity? Even in the absence of stimulus, the action potentials are generated in the auditory nerve. However, these action potentials, the number of action potentials that are generated during the silence or when there is no external stimulus, is called spontaneous activity. These numbers are very low, and also these numbers are random. That means there is no particular pattern in which the action potentials happen during the spontaneous activity. It is said that this randomness in the action potential typically codes silence. This change in the spontaneous rate of the action potential might trigger the tinnitus, and this change in the spontaneous rate of the action potential might happen because of any of the tinnitus triggering mechanisms such as hearing loss or the noise exposure or the anxiety or ototoxic drug, all of this. So what are the different types of changes that can happen in the spontaneous rate? The spontaneous rate can increase, or there could be increased synchrony in the spontaneous rate, or there could be bursting of activity of the auditory nerve. So increase in the spontaneous rate in animals where the tinnitus is induced is seen in auditory nerve, has been observed in dorsal cochlear nucleus, has been observed in inferior colliculus, and also has been observed in auditory cortex. The increased synchrony, I said the spontaneous rates, generally they are very randomly spaced. There is no particular pattern in which it's happening, but because of certain of this triggering mechanism, these spontaneous activities, because of certain changes that happens in the cochlea or in the outer hair cells, may pick up a particular pattern or happen in synchrony. That is, there is a lot of auditory nerve fibers might first start to fire an action potential at one particular time, causing a synchronous firing of the action potential. Because this firing happens synchronously, the auditory nerve, the brain or the auditory cortex might perceive this as sound, because earlier there was no synchronous firing, it was happening randomly, and suddenly the pattern of the action potentials changed in between, and the brain might start perceiving it as, and might give a more perceptual weight and might perceive that as sound. So this has been observed in both noise induced tinnitus and also in the salicylate induced tinnitus. It has been observed in auditory nerve. It has also been observed in the inferior colliculus and in the auditory cortex. And the bursting of activity is also observed where the output of the auditory nerve or the cochlear nucleus neurons are at a particular instance of time, there is a burst of action potentials coming together, and there is a silence for a while, then again burst of action potentials coming out, and there is a silence for a time. This is also seen in animals where they are exposed for the noise or treated with the salicylate for inducing the tinnitus. So this is about the neurophysiology of tinnitus. There are few models that have been proposed for the tinnitus. One of the very popular model is the central compensation. Now, this is based on a concept called homostatic plasticity. Now, what does this homostatic plasticity means? The auditory neurons adapt to maintain a network of similar to before peripheral hearing loss. That is, if there is a certain output from the cochlear nucleus or the certain output from the inferior colliculus, okay, the auditory neurons, the auditory network in the brainstem and the cortex is programmed in such a way that irrespective of what is the input, the output from these neurons are programmed to maintain in a certain way. Now, in normal conditions, there is an input from the cochlea or the auditory nerve, and there is an output, a particular output that's coming out of the cochlear nucleus or the inferior colliculus. When there is a hearing loss, what happens is the output from the cochlea is reduced. Because output from the cochlea is reduced, and the structures in the brainstem and the cortex will maintain homostatic plasticity, that is, they will maintain certain amount of the output irrespective of what is the input, and because of the hearing loss, the input is reduced. Now there is a compensation, or there is an increase in the output of the cochlear nucleus or the inferior colliculus neurons. This is called overcompensation or maintaining the homostatic plasticity. So in animals who are treated for the tinnitus, in whom the tinnitus has been induced with the salicylate or with the noise, people have observed increased neural activity, especially in the dorsal cochlear nucleus or in the ventral cochlear nucleus and in the inferior colliculus. Typically, typical expectation is that when there is a hearing loss, when there is a lesser input, the output also should be less, because the cochlear nucleus or the inferior colliculus is not receiving an input from the cochlea. However, that is not the case. Because the input is less, these structures will try to compensate by increasing the gain or increasing the number of action potential. So this would result in larger output from the brainstem structures such as cochlear nucleus and inferior colliculus, and this has been experimentally seen in this structure. This is one such example of increased activity of the neurons in the cochlear nucleus cells following a noise exposure, and this following the noise exposure, and the animal did perceive behaviorally the tinnitus. Over here there are the graph is very complex and has a lot of information. Let's see one by one. The gray color one, what you are seeing over here, is a noise to which the animal was exposed. So this is the spectrum of the noise. So you can see that the noise had a peak somewhere around 6 to 8 kilohertz and had a lower energy in the low frequencies and had a higher energy in the higher frequencies. So if a person or an animal is exposed to this kind of a noise, typically hearing loss will happen 1 to 1.5 octave above the peak of the spectrum. So if this is the noise peak of the spectrum of the noise, typically hearing loss should happen somewhere around this region. So the filled boxes over here, the filled circles over here indicates the APR threshold of the change in the APR threshold of the animal. So you can see that 0 means there is no change in the APR threshold. Now, for example, 30 dB means the APR thresholds are increased by 30 dB compared to pre-exposure. So you can clearly see that the APR threshold in the lower frequency regions are less, and APR threshold at a frequency which is slightly 1 octave or half octave above the exposure frequency is almost 50 dB. There is 50 dB hearing loss caused by this noise exposure in this animal, okay, at these frequencies. And this shift is temporary, because when the same thing was evaluated after a week, all APR thresholds have come back to normal, or the shift is 0. So temporarily there is a damage to the cochlea and probably to the spiral ganglia neurons, which is causing the elevation in the threshold shift in these animals. Okay. But what is interesting is the red curve, which shows the spontaneous discharges of the neurons in the cochlear nucleus. So you can clearly see that this is shift in the spontaneous discharges in the spontaneous discharges of the cochlear nucleus neurons. What does this show? When this red curve is nearly 0, when the red curve is at 0, it indicates that there is no difference in the spontaneous discharges between pre-exposure and the post-exposure. Over here it shows that, now you have to see this axis. Now over here it shows that at this frequency it is around 15 spikes per second, which means the spontaneous discharges were 15 spikes per second less pre-exposure, and their spontaneous discharges are increased by 15 spikes per second in post-exposure. And at the frequency which is very near to the tip of the spectrum or the highest peak of the spectrum, you can see that the spontaneous discharges are increased by as much as 50 spikes per second, which means that this particular neuron before exposure, if it was firing at a rate of say 2 action potentials in 1 second, after the exposure of the noise, this particular neuron will fire at a rate of 51 or fire at a rate of 52 action potentials in 1 second. So that means the spontaneous discharge, even in the absence of any sound, the output of this neuron in the cochlear nucleus has increased primarily because of the exposure to the noise. Okay, though the noise has caused hearing loss, noise has caused peripheral hearing loss, it has increased the spontaneous discharges. This is kind of an opposing effect. At a particular when we give a sound, it causes hearing loss, or the person is not able to hear the sound. But in the absence of sound, the spontaneous discharges are increased. That is, the output of the auditory nerve in silence is enhanced, but output of the cochlear nucleus neuron for silence is enhanced, but output of the cochlear nucleus or the auditory nerve for sound is reduced. Okay, so in this particular experiment clearly shows that there is a very possible causal link between the discharge rates of the spontaneous discharge rates of the neurons and the tinnitus, where they showed that whenever there is a tinnitus induced, when we induce the tinnitus in the vicinity of the frequency range which induced tinnitus, the spontaneous discharges are much higher following the exposure of noise compared to pre-exposure spontaneous rates. The similar observations have also been made in the inferior colliculus. So this example shows the spontaneous rate in inferior colliculus across different regions of the inferior colliculus. That is, when the needle is moved from peripheral to the central, this is the depth of the needle. So you can see that all these are the pre-exposure spontaneous rates, which is almost close to 2 or 3 action potentials per second. However, post exposure, that is after the exposure to the noise, the output of the spontaneous discharges of the inferior colliculus has gone up to 20 spikes per second. However, they have also seen that this improvement or this enhancement in the spontaneous discharges are in fact driven from the cochlear nucleus. So if the cochlear nucleus is ablated, that is, in these triangles what you are seeing is the same animals who are exposed to the noise, but the cochlear nucleus is ablated or cochlear nucleus is cut off. When the cochlear nucleus is taken off, the spontaneous discharges in the inferior colliculus don't increase. When the cochlear nucleuses are intact, then only the spontaneous discharges in the inferior colliculus increases. In other words, the cochlear nucleus and the inferior colliculus are two main structures which are involved in maintenance of the tinnitus, or the brainstem structure, which are frequently cited as the location or the generator sites of the tinnitus. Though the tinnitus is generated because of some peripheral alterations in the cochlea, like because of the hearing loss or because of certain changes that happens in the inner hair cells or in the tectorial membrane, the discharge rates of these structures, that is the one in the cochlear nucleus and in the inferior colliculus, show an abnormally increased activity or abnormally increased spontaneous discharges in animals where the tinnitus has been induced. Okay, so that's about the neurophysiological some studies showing the different structures responsible for the tinnitus. I'll just touch upon the neurophysiological model for tinnitus, because the tinnitus retraining therapy that you would study, that you would hear tomorrow, is primarily based on the neurophysiological model developed by Jastrabov. This neurophysiological model was developed because a lot of people with a hearing loss hear tinnitus, but not everybody gets affected by the tinnitus in the same way, and there is hardly any relationship between the severity of the tinnitus and the behavioral consequences of the tinnitus. Means this is our I think a routine clinical experience where people who, some of the, when we assess for tinnitus using a loudness matching or a pitch matching, people may have a very high intensity tinnitus, but their reaction would be very mild. But there may be some people who have a very mild tinnitus, like a 5 dB or a 2 dB, 3 dB tinnitus, but their reaction is very high and their severity ratings on tinnitus handicap inventory would be too severe. So to explain this, a neurophysiological model was developed, which assumes that other than the auditory system, limbic system and autonomic system and emotional reactions also play a critical role in the tinnitus. Just to understand neurophysiological model, let's see, whenever the new sound comes, what happens typically in the auditory system. When the new sound comes in the auditory periphery, that goes to the subconscious level of processing in the brainstem and in the thalamic area. The parallel lead also goes to the limbic system and the autonomic nervous system to evaluate the emotional reactions to the sound. So any new sound which we have not heard earlier would be evaluated by a limbic system and the autonomic nervous system for the emotional reactions. The autonomic nervous system prepares the body for fight or flight. That is, if it thinks that the sound is dangerous, it will prepare the body to fight or run away. The limbic system will also prepare the body for adequate amount of emotional reactions. And it also goes for the conscious perception in the auditory cortical areas, and we hear and understand, and all of that will happen if the new sound is not associated with the significant emotional reactions. So if it is a familiar, if we keep on hearing the same sound and it is not significant, for example, a fan noise which you might be hearing now, or a truck which passes by the road every now and then, or a refrigerator noise in your house, you would be hearing it continuously. So it also goes to the auditory periphery, it also goes to the auditory subconscious, that is thalamus and the brainstem structures. Now it won't go to the limbic system and the autonomic nervous system, it won't get activated, because previously we have evaluated and decided that it is not important or it is not significant. So many a times this is blocked at this level itself, it doesn't reach the conscious perception, and we won't even become aware of such sound unless we pay attention to it. It happens a lot of time, like when you're listening to the fan noise or the refrigerator noise or even the road traffic noise. We are not aware every single time these things happen unless we pay specific attention to that. However, if the sound is familiar and significant, that is whether it is positive or negative, always there is some kind of an emotional reaction goes on building up before it goes into the auditory cortex. Now, the association that forms between the sound and the reaction is depends on type of the stimulus. At particular instance, probably there is no relationship between the stimulus and the reaction. That is, for example, say I'm sitting here and I heard a truck noise. There is no relationship between these two. These two just happen to occur, or happen to there is a temporal association between the perception and the reaction. There is a truck noise, I just turned. Other than that, there is no reaction, there is no relationship between these two. But if this is reinforced, that is negatively reinforced either or positively reinforced with a reward or a punishment, the reaction becomes stronger and stronger every time there is a stimulus. So we start to associate stimulus with a particular type of a reaction because it is being reinforced or punished. Okay. If this happens for quite some time, then even without the reinforcement, there would be reaction. Okay, that is, just the stimulus itself can evoke the reaction even without the reinforcement. Typically, in case of positive reaction, positive reinforcement, or in the pleasant situations, what happens if there is no reinforcement for quite some time, then the reaction will start deteriorating and there is no more reaction. Like for example, every time you hear a sound, if every time a baby, like in the visual reinforcement audiometry, what we do, every time a baby hears a sound and if it turns, we provide a positive reinforcement. So next time, even when you train the baby for this particular conditioning, next time when you just give a sound, baby would turn towards the toy or towards the monitor in anticipation of the reinforcement. So, but you don't need a reinforcement. Every time you present a sound, baby will turn towards the reinforcement. But if you keep continuing this for quite a while, what happens after some time, baby will realize that there is no sound. So it will, even if it hears a sound, it won't turn towards the reinforcement because the extinction has happened. That is what typically happens in a positive reinforcement or for a neutral stimulus. However, if the reinforcement is negative, instead of showing the baby a cartoon or a toy, if you give an electrical shock or if you hit the baby, there is a very strong association that develops with this negative reinforcement, and the reaction itself will act like a reinforcement, and the reaction keeps on growing. And this is what has been proposed to happen in the neurophysiological model in tinnitus. That is, you hear tinnitus because of some auditory peripheral abnormality, either in the cochlear nucleus or in the cochlea or in the inferior colliculus, because of some reason, because of increased spontaneous activity or whatever. it goes to your subconscious level, it goes to your limbic system. Once it goes to your limbic system, if you start associating negative consequences to this loop becomes stronger and stronger and stronger and stronger. Here the darkness and the width of the arrow shows the strength of the association. You can see that every time you hear tinnitus, you associate a negative reaction, and that vicious loop keeps continuing. And even though you get into the conscious perception, the limbic system and the autonomic system gets activated every single time you hear tinnitus. Because autonomic nervous system gets activated, you become restless, you become tired. That's most of the tinnitus patients will complain that they are very tired, they feel like running away, because it activates their autonomic nervous system continuously. And more importantly, at this point in time, the tinnitus itself, which is a root cause for all of this, becomes irrelevant. The strength of the tinnitus becomes irrelevant. Even if you suppress the tinnitus, or even if the tinnitus loudness becomes smaller and smaller because the pathology is resolving, even a very soft tinnitus will induce this cycle again, and the reaction would be very severe. So the initial sound or the tinnitus that induced this kind of a reaction becomes irrelevant, and the entire this vicious cycle keeps on continuing, and that will lead to a lot of other behavioral or other symptoms that are associated with the tinnitus. So this is the basic, in the nutshell, the neurophysiological model of tinnitus. And the tinnitus retraining therapy aims at breaking this vicious loop and helping the person, though there is a tinnitus, if this loop is broken, then he wouldn't be bothered by the tinnitus so much that he would be otherwise. There's another model of tinnitus which is very similar to the neurophysiological model called gating model of tinnitus, where assumes the same thing, that when we hear some sound from the cochlea, it goes to the auditory midbrain and the brainstem structures, and parallelly it also goes to the, from the thalamus, it also goes to the amygdala and subcallosal area to evaluate the emotional aspects of the stimulus. Now, if the stimulus is emotionally irrelevant, or if it is not a warning signal, these will send a inhibition signals to the thalamus, that is the thalamus and the reticular formation. And hence, if the stimulus is not emotionally relevant, or if it is not important, they are suppressed over here and they are not sent to the auditory cortex. Or even if the person is hearing tinnitus, there is a suppression of all this, what goes to the auditory cortex. If there is a problem, if this structure is compromised, the subcallosal structures are compromised, the long-term habituation or the inhibition will not come into the thalamus, and hence every time the person will hear the tinnitus and he will get bothered by the tinnitus. So this is, they call it as a tinnitus. This is called compensated tinnitus, because even in this condition, the tinnitus is present, the tinnitus is not gone, just because the subcallosal structures and amygdala, which is responsible for assessing the emotional content of the emotional content of the input, is working fine. There is a inhibition. This results in the long-term habituation, and the output from this thalamic nucleus will get suppressed. You don't perceive it tinnitus in the auditory cortex consciously. If these are compromised due to some illness or some disease, or because of some any other reason, the long-term habituation doesn't happen, so the tinnitus will always be perceived in the auditory cortex. So all this model will help in understanding the tinnitus, as well as trying out different treatment technique, either cognitively, behaviorally, or pharmacologically. Like for example, this particular gating model of tinnitus has helped in finding out certain drugs which would increase the activation of these subcallosal areas of the brain, so that the long-term habituation can take place and tinnitus is no longer in the conscious perception of the auditory cortex. Okay, so this is about, in briefly, about pathophysiology and certain models of tinnitus. I would like to thank organizers for giving me an opportunity to talk to you all about this. If there are a few questions, you can probably type in the chat box. We can try to answer, or we can get back to you if the time doesn't permit.
Thank you, sir, for the informative talk. Now I will be posting a link having the questions to be answered by those people who want a certificate telling they participated. Also for those people who are requiring RCA CRE credits. So I'll be sharing the link. You can click on the link and you will have 10 minutes to answer. So after that 10 minutes, the link will not be active anymore. So in 10 minutes, you will be having to answer 5 questions only. It's an objective kind of questions. You have to just click from 4 options given for each question. If others also, if who are interested to take the test to understand how much they've learned from the lecture, they are also free to do it. But people who are requiring RCI CRE credits and requiring a certificate telling about participation, they should be attending this. So I'll be posting the link in the chat box. So you please, I am sending it right away. And others, in the meanwhile, if anybody is having any question, please do feel free to type it in the chat box. So the link is there in the chat box. You can just click on the link and start answering within the next 10 minutes. Or it is like once you open the link, 10 minutes you will have to answer. Thank you.